Hello. How are you?
Good morning.
Morning. All right. Welcome. So yeah, Jesse is taking leave of absence from the lab. He's busy with things in his life. So
Finishing up as being a student.
oh yeah. So yeah, that's good. And we were going to have a cybernetics meeting last week, but we postponed it for the week after. And I think we had our third DevoWorm meeting last week, and we covered two papers. And so one on benchmarking C. elegans data sets, which was really interesting. So if people are interested in that for like machine learning or whatever, they have some benchmarks in there. We went through their GitHub repository from the paper and looked at the different benchmarks that they have for, I think there were some behavioral benchmarks, some cell biology benchmarks, and things like that. So that's interesting. Another thing that happened was I got the project descriptions for Google Summer of Code to INCF. They're kind of taking a first pass at it, so I sent in some of our ideas, and hopefully we'll hear back in a couple weeks about the status of that. So did you have any updates you wanted to share, Morgan? Or?
I've got two dogs here I need to deal with, but I've been doing Josh Bongard's course. Well, I've been attending all the lectures for the Evolutionary Robotics class.
Yeah.
And that's been interesting. I'd like to follow up on that. This dog wants to go out. We did a reading group on the active inference textbook.
Oh yeah. Yeah.
And so three people showed up.
Oh wow.
10 people were signed up.
Oh.
And you know, the vehicles one was much better attended. You know, I just don't know if you can just give people the entire 300-page book as a
yeah,
even with kind of 2 months notice.
Yeah, I think that's kind of like, here's a book, you got to go chapter by chapter.
I know. Anyway, and but by three people, I mean I was one of the three. So there was two other.
Yeah,
well, there was like the guy. Yeah. So I definitely shared that there were other ways to do this, and the Active Inference Institute is the is the master of this. As well as sharing the step-by-step tutorial, and the fact that they've got like almost 8 hours of video talking with Ryan Smith and Christopher White about the step-by-step tutorial. So that yeah. I can say more about the robotics in a minute.
Okay.
And attended the 3D slicer. It's not. They called it a hackathon, but it was work on a project. It was like developer day. Yeah. So that was cool, and I was just kind of focused on what we'd discussed, like what was that, 2 weeks ago now, a week ago, um, just about you know the issues building on Fedora, and yeah, so interesting stuff where it's like I actually did make progress. And but it's silly things like you know oh hey I've got to use an older version of GCC because like too many what would be warnings on GCC, like they're building on Ubuntu,
Yeah.
and Ubuntu's like 3 versions behind Fedora in terms of GCC.
Yeah.
And anyway, so it's funny when you realize that it's not version issues. It's not like code issues or anything like that. It's just that yeah, you've got to downgrade your system so that the builds will complete.
Right.
Um, but that was good, and it was nice to connect with those guys, and uh, yeah, like their kind of open source community manager was there helping people. They were doing an in-person event uh, in North Carolina,
Okay.
and and uh, right now is BrainHack Vanderbilt, um, which we could actually uh, maybe could check the talks online.
Yeah. All right. So there are a few things I wanted to go over. I think some of them Morgan had mentioned, and then I think one was where Hussein is involved. So the first one is the Slicer Hackathon. So this is hosted by Kitware. This is this page on the 3D Slicer community. So we've talked about 3D Slicer both in this group and in DevoWorm, where people are doing 3D modeling of image visualization, analysis, and there's a wide variety of people in that community, including 3D printing people, paleontologists, astronomers, surgical people, medical imagers, etc., etc. They were doing this in-person event at the Kitware North Carolina office, and they also, I think, had a Google Meet instance where people could join virtually. So this is a nice example of an event that people host online, sort of in this case, hybrid, and they're able to kind of organize this on their discourse instance. It's like this is a typical hackathon where they want you to review the issues, set up your environment before you get there, and then get into these discussions about different topics. So their good first issues are here on their GitHub repository. These are just basically very simple issues. Simple if you're in that community. If you're, a very early beginner, probably not. You have things like improving the developer's guide, document the slab reconstruction UI, documentation of CMake variables, improve slicer execution model documentation. These are things you might expect from good first issues and very simple issues to sort of solve during a hackathon. You don't want to have huge issues during a hackathon. You want to start small. Maybe if you decide to build something, you kind of figure out what the issues are and attack the low-hanging fruit. And then after the hackathon is the time where you really kind of move up to the more complex issues. Yeah, so that looks really nice that they had this event. Looks like their community is well structured, and yeah, that's great.
You had a pretty sizable group there. Sorry, I'll try and find some lights here. There in North Carolina.
Good.
Yeah. Ooh.
Oh,
Sorry.
there we go.
And yeah, I'll figure out how to kind of give them a report or it's a big project, like they have a lot of dependencies, and like CMake the build tool started because kitware, it was developed, in the perfect kind of hacker way, where the developers needed to scratch their own itch in that they needed to build VTK across multiple platforms, right?
Here.
So there, it's like we need to support Windows, which is always the big problem.
Yeah.
And Unix.
Right.
And yeah, and then the fact that like all these platforms have different different tool chains, so anyway, so I saw a lot of those issues, but also just like that you're building. One of the things that Fedora dislikes, but is very common, especially with scientific software, are these super builds where instead of using the system dependencies, instead of using the system builds of various commonly used tools, you build locally every single dependency you have, which makes for just really long build times, as well as like a lot of redundancy in terms of just everybody's building their own ITK, everybody's building their own VTK, instead of trying to figure out how to use the system version or something like that, for instance.
Yeah.
But that was exactly what I knew was going to happen, and so I got to learn a little bit about how to better adapt to the system. I was still using the super build, but I was turning off tools that were that I had obviously as system dependencies, as well as like figuring out that I needed to use GCC 13 instead of 14. Which, again, these are the kind of things where it's just like, hey, for some reason GCC now throws errors instead of exceptions. Instead of warnings for exactly the same code. And so, but that was good.
Good. Yeah.
Yeah. You're going to talk about the other hackathon?
Yeah. So it looks like Hussein went to the MIT Reality Hackathon. We talked about this last year. It's from 2025. It's for 2025, and I don't know if they're different things going on here. It's basically the same structure, but so yeah, they had the 2024 tracks and prizes, and so let me see if they have anything in the experiential 2025. And I don't know if they tell you much about. Okay, here it is. The program structure. So this is the technology theme is deep spatial, the next generation of extended reality, and they have extending reality and all these application domains, designing prosperity, learning innovation, advanced computing, integrating embedded systems, hack to market, which I assume is like just creating like rapid prototypes of things. Engineering sustainability, multisensory interaction, digital embodiment, aerospatial applications. I'm not sure what that is. And then artificial intelligence. So this is just kind of like the tentacles of extending reality. So there are three categories: technology, methodology, and action. So technology is experiential now and soon. So that is what technologies are experiential now and in the near term. What are they, and what potential implications do they have for creating new kinds of technology-enabled human experience? How is this technology currently being enabled by other institutions for world development? So I guess it's just putting the technology into practice and how they sort of enable human experience. Methodology, category 2, how to foster innovation. So this is just kind of how do you make things. One core hypothesis behind the reality hack mission is that forms of technology development leave gaps in what problems are solved and who is served by new technologies. What relevant methods and mechanisms have previously been proven successful for shepherding innovation, from enabling would-be innovators with skills, self-efficacy, and a support network of people and resources to deploying commercially successful solutions that solve problems and make the world a better place. Is that a question? I don't know. What new mechanisms might be conceived and implemented? So this is where you're enabling people, giving them the support to create things and put them out into the world, and then also making sure that those things are useful, or enrich people's lives. And so, this is always something that people are looking to do, is to figure out how to foster innovation. And then 3 is action. This is Let's Reality Hacks. This is just kind of like doing the things you do at a hackathon, creating your project, doing, collaborating with other people, doing things in person in a short period of time, and then producing some long-term project. So this is pretty nice. Hussein's really excited about it. He's always excited about it. It's a nice event. It's, I think, at MIT, and the salon at MIT, Green Building, MIT Museum. So yeah, it's nice. Let's see if they have any more information about what's going on. So it looks like they have Salon 1 opening kickoff state of the industry. Little Hands, Big Worlds, XR for Kids at the Yale Center for Immersive Technologies in Pediatrics. This is the 1035 Startup Talk, Hapticos product launch. I guess this is a haptic device. Then I suspect that the rest of the session is like, kind of getting into the hackathon.
Mm-hmm. Yeah, I know OpenBCI is there because they've got that Galia headset, which is, a VR EEG.
Oh, right.
Integrated device. I mean, I think it's got some additional features as well. It's a very spendy device.
Yeah, I know.
20, 25K, 30K.
Oh, wow.
Yeah. I mean, that just tells you how much. Excuse me. That tells you what their development costs would be. I mean, like the hardware doesn't cost that. I mean, the bill of materials is not that expensive.
Right.
But the need to recoup the engineer time, basically, that you've got on that was apparently significant. But yeah, that I've seen OpenDCI posting stuff from the event. They were talking about it before too, and of course, yeah, you're saying it's very. I mean, he attended last year too.
Yeah.
Yeah.
Okay, so that was the MIT Hackathon. Then something posted by Morgan, I think it was in one of the, I think it was either in Brainbird Vehicles or DevNeuro AI, was the main conference playlist.
Yep.
So yeah, this is the videos that they have, 27 videos. So it looks like they had opening remarks, aligning representations across individual models and naturalistic neuroimaging.
She's a staple of the open source, brain hack and other communities.
Elizabeth Dupree,
Yeah.
yeah.
Arena's obviously a professor there at Montreal.
Yeah.
And well, I guess obviously we all know Josh.
Yeah.
I think Elizabeth is she Stanford now? I forget. Yeah.
This Arena Risch talk, rethinking intelligence from dynamical systems perspective. It's interesting.
Yeah. I mean, Terry Stanowski's obviously, is the one who, every time I think I find some interesting new math physics thing, it's like I'll do a search, and Terry Stanowski described it 10 years ago.
Yeah, he's been doing a lot of this for a very long
time. So I wonder what kind of overlap there is there, but yeah, and I guess eventually he needed to talk about transformers.
Yeah.
But I just saw a talk on just how crap transformers are for time series prediction.
Oh yeah. Yeah, any kind of sequential data, I guess, is the problem.
Yeah, it's yeah. Just treating time series as a token prediction is just not.
Yeah.
But he's the master of I think it was these delay differential equations that I started seeing like last year, and he's got a paper in 2013 about them.
Oh yeah, they've been around for a while.
Oh yeah, I mean they have, but just applied to neuroscience.
Oh okay.
Yeah.
Yeah, I don't know what. Yeah, it'd be interesting to see what he has to say about some of this. And the panel discussion: does better AI mean more human-like AI? This one here: 2 new perspectives on the structure-function relationship in neural networks. Paul Cizek, who we've talked about, some neuroscience foundations for AI. The second keynote is Luis Pessoa, the entangled brain, integrating perception, cognition, and emotion. Then there were some lightning talks.
Oh, okay. Entangled brain guy. Yeah.
Oh, this,
I mean,
the Luis Pessoa, yes, entangled brain.
Yeah.
I take it that they're getting, like, a lot of this is at the level of, here's my work, how does that fit into the greater vision? Which we might talk about in the panel.
Yeah.
Experimentation on in silico neural responses from encoding models, on continual reinforcement learning, doing a pre-cook. Then the next panel, emergence of reasoning in brains and machines. Some other talks here on, I guess, visual representations, visual sensation and perception, visual processing, on the relationship between single units and distributed representations.
That's interesting.
Typical sort of like visual representation stuff. Another round of lightning talks. Foundation models for neuroscience.
Yeah, he's now paid by the Amarath Foundation.
Oh yeah.
And they're doing the Enigma, the unfortunately second project to be called Enigma.
Yeah.
And well, there's also the big psychiatric one,
Yeah.
It's kind of like a single unit. It's like a foundation model from the ground up, like using big new data collections, I think is the idea.
Yeah.
Excuse me. So idea being that, like, or I don't know if this is how they justified the funding for it, but the brain project, the brain initiative is being defunded. Somebody needs to do this right.
Yeah. It's always 10 years later, do it right. You do a project. Well, we got to do it right this time.
Maybe. I mean, together with this hype about foundation models.
Oh well, yeah. I mean, I wonder if they're dealing with that any better than anyone else. Like, it seems like everyone has a foundation model for something.
I mean, I love him. Like he's always been attached to really cool, good, I mean, he was CTO for Neuromatch. He was, it's good neuroscience, Mila kind of projects. But there's just some of that. He's at Foresight Institute meetings.
Yeah.
And there's just this whole brain emulation. We should did I put this in? Carbon Copies is doing something big February 22nd. Did I drop that in call for participation? Sorry, let me.
Yeah, Carbon Copies Foundation on LinkedIn. Brain Emulation Challenge Workshop.
Yeah.
Okay.
Yeah. And again, there's. I think I've said before that there's some discussion around whole brain emulation that I just find. I don't know what the right word is. Like, it's not just that it's optimistic. It's like it's wishful thinking.
Yeah,
it's like wishful thinking around this that doesn't seem to
match reality, or what
doesn't seem to be well grounded. It reminds me of the Far Side cartoon where there's a lot of equations, and then there's like, and then a miracle occurs.
Yeah.
Anyway.
Well, I mean, it's certainly like an open set of open problems that, like, if you think, okay, well, we have this vision for something, and we have the state of the field, and then there are like 5 big open problems we need to solve to get to what we want to do. And it's great if we could solve those problems, but we're not there yet. So let's just kind of work around that.
Anyway, sorry, I was just going to say something about carbon copies where, there's a lot of really good technical, he and Logan Collins have a really nice white paper discussing the technical innovations that would be that are within reach and practical, and what they would do to improve the just our ability to capture connectomics and cell structure.
Yeah.
That's really interesting, and yeah. Anyway, and I just saw that Philip Shue, who was the first author of the big Drosophila Connectomics gives us behavior paper. I don't know if you remember this from Kristen somebody's lab in Berkeley. Sorry, but I can find Philip Shue. I can find the link. But it was an interesting, like, hey, connectomics gives us something akin to connectomics plus computational neuroscience simulation gives us something that looks like calcium imaging.
Oh, really?
Of the same stuff.
So, like, this is just like a standard simulation, and then the connectomics data on top of that.
It's connectomics data, and again, like, devils are in the details. Devils in the details in terms of just like, okay, like, does it? I think that, like, I think it's one of those things where, so it was like, this simulation gives us something that looks like olfactory, olfactory response or something like that. But sorry, talking last night with my daughter about 23andMe results.
Okay.
Where it's just like, hey, she's saying something like, what's my ancestry? It's like her mom's Irish and I'm pretty much English, and so it's saying, well, according to 23andMe, I come from London. But here's the thing, right? I was trying to explain to her that, this kind of DNA analysis, it's not like they go in and they like, oh, you have London.
Yeah.
You have London in your DNA, right? Like it's on the basis of who signed up with 23andMe and where are they located. And so you have this problem that in the UK, of course, like, most people live in London.
Right.
And certainly people with disposable income to waste it on a 23andMe kit also live in London, right? So it's just like, do I come from London, or is all the samples just from London, right? Well, it's did the connectomics results give you an olfactory response on the basis of connectomics?
Yeah.
Or is that just like, is olfactory kind of like the main sense? Is that like the visual system for Drosophila?
Right.
In the sense of like you stimulate somewhere, you're gonna get the biggest response in olfactory areas or something like that. You know what I mean?
Right.
Anyway.
Well, that's an
This is,
interesting. Yeah, that's an interesting question. Is that like an issue of sampling then, or is it like?
you know, I don't want to, you know, I yeah, I mean, it would be great to have somebody like Philip Shue address that.
Right.
I will point out that one, um, he was totally not interested in organoid intelligence. And maybe I'm still feeling hurt about that. And two, he's currently working at, I forget what they're called. Maybe it's like Emulate Bio. Anyway, he's working at a startup downtown that is doing whole brain emulation.
Oh, okay. Yeah.
You know, like which to me means that they're doing histology and find high res. You know, I mean, they're basically they're doing histology. You know,
Yeah.
But it's probably easier to raise money with whole brain emulation. But,
yeah, it's sexier, as they say.
Yeah. Again, I still want to know how they think that what's the secret sauce?
Yeah. So that was the Patrick Minialt Foundation Models for Neuroscience. There's another talk here: a common neural basis for behavior across individuals and species, which is interesting in light of all the model organisms we use, but also in terms of the phylogenetic approach to neuroscience and how we can sort of extract key principles across different model systems. Blake Richards had a talk, Brain-like Learning with Exponentiated Gradients, talking about some of the problems with gradient descent and getting at those kind of issues. Then this keynote here, number 5, Laura Driscoll, Fast and Slow Learning in Artificial and Biological Networks. The panel discussion for day 3 are foundation models for neuroscience, the next big thing. Obviously, that's a contentious thing. Then the last few talks here, multi-agent cooperation through learning aware policy gradients, more stuff on gradients, credit assignment in neural networks without plasticity, and then large scale pre-training on neural data allows for transfer across individuals.
She's the one. Is she at Emory?
Ava Dyer, maybe.
Yeah.
Okay, and then there was a
great meeting.
Yeah, closing address here, and it's a great meeting. And check out some of these talks. It's on the main conference YouTube playlist.
Yeah. I mean, it's like how long they've been going now? 6 years or something. I mean, they've been going a long time. It's great. Great meeting. One of the only ones that really centers itself around neuro AI.
Yeah. So we might get into brain organoids. I don't think we've talked about brain organoids in a while. Kind of some of the things from I think November to now. So let's start with the first thing here. This is this LinkedIn link about Assembloids. This was Sergio Pascal on LinkedIn Assembloids, and is showing this diagram here where we have neural organoids, different types of neural organoids, different, striatal organoids, spinal cord organoids, muscle organoids, glioblastoma organoids, combining those into neural assembloids, which are these hybrid things here in the middle, and they innervate each other, and then disease models, which are the migration assembloids, creating different tissues, or sort of, you can have like mutations, or you can have different disease states. So, for example, glioblastoma cortical assembloids can be used for cancer invasion models for tumor cell metastasis.
or the way Open Water was using them for demonstration of your ability to destroy glioblastoma and leave healthy cells.
Yeah.
Pretty remarkable.
Yeah. So they have they have two different types of neural assembloids: migration assembloids and projection assembloids, and they involve different combinations of organoids. So, like, some are where the cells migrate from one to the other. You have two organoids put together. The cells will migrate across, and then projection assembloids, where they'll send projections across, and that's the dominant form of integration. And so then that allows you to study other types of connectivity defects as opposed to migration defects, where you're looking at things like neural degeneration, reduced network synchronization, and abnormal neural activity. Okay, that's a great diagram. I think we saw this before in another meeting, but I wanted to bring it up. It's a nice diagram.
Yeah, he keeps on. This is one of Pasca's particular, hammers, if you will.
Yeah.
Is Assembloids. I'd still probably credit is it Knopfler? I'm trying to think of the guy in Vienna who was Madeline Lancaster's advisor. She's the first author. Interesting. There's a guy on LinkedIn who's been trying to do summaries of organoid research, and he obviously put forward some frontiers paper image that had a table like this, but had like kind of an obvious mistake in it, because it was talking about a whole brain organoid.
Yeah.
And anyway, but it was interesting because people like Allison Mutri down in San Diego chimed in about both mistakes in the figure as well as like papers that they felt were, I think, because he was saying something like cerebral organoids were first first reported in 2013 or 2012, 2013, something like that. And it was interesting to have people like Mutri and Pasca jumping in on this, saying like, no, it's like 2008.
Yeah.
And yeah, so interesting. Yeah. Anyway, the Assembloids, fell down a little rabbit hole in terms of just what really constituted the first. I guess the other question was like human cell organoids, and then then the next question would be who started the Assembloid kind of craze.
Yeah.
Or connecting different organoids together.
Yeah.
But Pasca is of course doing the class doing the workshop that a lot of his posts relate to the upcoming workshop in April. So that's his brainorganogenesis.org. And still really hoping he'll make all those training materials available.
Yeah.
So definitely want to talk about this paper from 1 second. I don't think I put this in Slack, but let me just drop this in. So, yeah, there you go.
Okay. Yeah. So this paper here, this is an archive paper.
Yeah.
Oh, this one, yeah. Starting a synthetic biological intelligence lab from scratch. Yeah, I saw this was put up on the archive over Christmas break or holiday break.
Right. Right.
Yeah.
And you got a combo of people from both brain engineers, right?
Yeah.
As well as cortical labs.
Okay.
You know, so that was nice. And then I hadn't seen people from RPI before, but you know, more the merrier. And yeah, that was cool. And if you go through a little bit, especially to the supplementary information, it really does, you know, detail like what you need.
Oh yes.
Yeah. So this is you know, again, it's just hard to get overviews of what equipment do you need, what, yeah, like if you really just want to focus on growing neurons, doing recordings, and you know, and potentially interacting in this kind of organoid intelligence way. I kind of prefer this synthetic bio intelligence term, but anyway, really nice yeah.
So they're an organoid lab. I mean, or is this
you
more
got
just?
Sorry, go back up to the top. Yeah. So you've got Mustajo Raji. He's like, you know, I forget his exact role, but you know, he's definitely a senior PI at the Genomics Institute,
Right.
and of the brain engineers in particular at that, you know, because that's like brain engineers are like a sub organization of Genomics Institute.
Right.
I was surprised that David Hostler is not on that. Anyway, but that doesn't surprise me that Muhammad is, because he's on all the kind of the real organoid papers. And then Brent Kagan, is obviously the cortical labs guy. So I don't recognize anybody else. But I will see what I can learn about what's going on. I thought it was just interesting as well, the kind of do you see the design lab at Rensselaer?
Oh yeah.
You know, the Center for Biotechnology and Interdisciplinary Studies, which I thought was interesting too. Anyway, so I'm hoping that just means, you know, we've got this Organoid Intelligence Slack, and you know, trying to build a community of the groups that are doing this, right? And so maybe RPI is a new once you know, as a lab that is interested. So I guess the Gay Wang, I don't know how you'd say G. Gay Wang.
Yeah.
Let me see if let's see what I get through. And there's this new, this is the, sorry, I dropped this to, like you said, like there were a bunch of papers that that were dropped over the holidays.
Yeah.
And this is the one where, like kind of updates some of these, somewhat previous reports from the same group, but again, just touting their cloud-enabled organoid lab as being a kind of an empowering remote lab, right?
Yeah.
That you can work with this. And this is so it's just nice to see. This is what we're trying to, in terms of anything we build would be basically for others to use, right? And yeah, so I thought a nice report on that.
Oh, that's
and
great.
again, with Neurotech X, we've got some groups that are remote that that we're hoping that they can get access to the Braingineers work.
Yeah.
Want to bring that up, and oh yeah, and then the only reason I'm bringing it up is the connection with let me just see. Go. Let me just see if I get this with, yeah. And then the last I want to just bring up, because it comes back around to active inference.
Okay.
Or it could come around to that, but it's doing something that is dish brain. This is the first kind of dish brain like paper that I've seen from the Braingineers, and you can see Katerina's the middle author there, and then you've certainly got the usual suspects in terms of the Braingineers PIs at the end there. Yeah. So Mohammed, David, and Marcia.
Yeah.
And that is brought to mind. Just one sec. Got too many papers here. But a paper from Friston plus some other co-authors on active inference and intentional behavior. And the last author is Maxwell Ramstad.
Okay.
Anyway, I there's some, these two areas are kind of coming, starting to merge.
Right. That's great. So this is where they're kind of using cortical organoids to look at intentional or goal-directed learning. And then they have this part here. Although sensory input is vital to neurodevelopment in vivo, these studies have explored the effect and meaningful input to in vitro neural cultures over time. So this is where they're outside the brain and, giving input to developing organoids, I guess, organoid cultures.
Yeah.
This is the demonstration of the first example of a goal-directed learning observation in brain organoids. They have this closed-loop electrophysiological framework to embody mouse cortical organoids into a simulated dynamical task, which is the pull balancing cart pull task, and then evaluate learning through high-frequency training signals.
Yeah. And now if you just check the Slack again.
Okay.
And yeah, check there. So
There we go.
you see in their formal account of intentional behavior, we simulate in vitro experiments in which neuronal cultures spontaneously learn to play Pong.
Right.
So they're absolute. You know, it's this merging of the simulations of neuronal culture and actual organoid intelligence.
Yeah.
and it's the third leg of this, which I thought was super interesting. Actually comes from Josh Bongard. And he has he did a talk not as part of his lecture series for Texas Tech, and in this he is I'll drop it into brain organoids because it's again it sort of relates, but sorry, where is it? So it's at like 40. Yeah, it's about 45 minutes in. He is. Oops, sorry, I lost you. He's talking about the. So here's a class about evolutionary robotics where you're making simulations and you're designing, policies for these almost random bodies, right?
Yeah.
And they're mostly. I mean, it's mostly moved to soft body robotics at this point, right?
Yeah.
But yeah, the very crude. What I mean by that is like these are very crude structures,
right?
That don't certainly look like robots. But it's interesting because as soon as he gets to kind of his xenobots, he's talking about how they develop these with basically micromanipulation of embryos to get these kinds of behaviors. But then, and interestingly, talks about the fact that the word robot comes from this Czech word from the 1920s, I think. And in the story, these were biological robots.
Oh.
So he's just like, in many ways, these are robots. And the mechanical ones are the ones that are, are kind of fake version of them.
Yeah.
But he starts to do simulations of xenobots.
Okay.
And or simulations of what if I do micromanipulation of embryonic cells to create sheets of skin cells. I think he was mostly playing with, again, it's the work of somebody. Patrick, I forget now.
Yeah.
Patrick, making, fabricating cell structures of particular cell types, cardiac and skin, where his simulations start to make good sim to real transfers.
Okay.
Which is cool, right? So again, one of the things that he talks about in his class is the difficulty of sim to real transfer when they're just talking about the kind of soft body robots that they've been the kind of things that he's been working on for 20 years.
Yeah.
But what was really interesting was, hey, we're doing simulations of xenobots, and it's actually making useful predictions about what kind of cell structures we can put together and what their behavior will be. Yeah. So it doesn't quite come fully back to the Ramsted's active inference modeling of neuronal structures and the goal-directed learning of the Braingineers, but I'm feeling really good about that. Those three coming together, where where those simulations can get much more biological, and the reason for that, right, is that geometry and kind of connectomics, again, like one of the things that we haven't been able to do in organoid intelligence is, well, there's a great deal of trouble generating 3D structures that have purpose.
Yeah.
Or certainly just being able to control that in a again, a kind of experimenter goal directed way. Like I want to have this kind of brain structure. How do I signal that, right?
Right.
And I thought it was interesting that Bongar's not quite there, but he's predicting mobility off of these artificial, off of these biological robots.
Yeah.
So really, really interesting kind of confluence.
Yeah, I like this. Yeah.
I think I feel like I should use some fancy words.
Confluence.
No. What's the sociobiology guy?
E.O. Wilson.
Yeah. Consilience.
Consilience, yes.
He's got that book on consilience.
Yeah. Well, that's great. Yeah, thanks for that overview of things there.
Yeah. Sorry, not. There's some good stuff there too in terms of high throughput culture work, and there's I got some links to some what I would call kind of technical organoid culture papers.
Right.
Our messages are going to be deleted January 28th.
It looks like the older ones. I mean, the ones that as they disappear. I'm backing up these different channels in the Discord, so we have the links preserved somewhere, and I've also backed up some of the channels.
Okay.
So it's not going into the ether.
Okay.
But yeah,
it would be sad.
Yeah, I know. Why don't we move through some of these other links, and then we'll. So this is this is a tissue talk, I guess, that happened.
Yeah.
Programmable organoids guiding cell fate with synthetic biology and neuromorphic circuits.
Yeah.
And then
he's one of those people who's been doing it forever.
Yeah.
Yeah.
Sergiu Pasca.
Well, the Ron Weiss.
Oh, okay. Yeah. Okay.
The programmable organoids. Sorry, that was a different. That was tissue talk. Yeah, old tissue talk.
Yeah. And then we also have this paper here, and this kind of fits into what you were just talking about. Assembloid models to study loop circuits of the human nervous system. So this is where we have neural circuits that kind of loop around from the basal ganglia, the cerebral cortex, and the thalamus. I mean, these are motifs that you'll see in the human connectome, and oftentimes those are, important for function between, like, things coming up from the brainstem to the neocortex and then back around and looping. So this is basically, the inspiration for the fundamental networks for sensory motor processing and their dysfunction, so you can study neuropsychiatric disorders. These recursive loop circuits have been investigated in animal models and by clinical neuroimaging. However, direct functional access to developing human neurons forming these networks has been limited. So, we can use animal models in mammals to look at these circuits. Here we use human pluripotent stem cells to reconstruct an in vitro corticostriatal-thalamic cortical circuit, which is this loop between the thalamus, the striatum, and the neocortex, by creating a 4-part loop assembloid. So that's the 4-part loop assembloid.
Yeah.
More specifically, we generate regionalized neural organoids that resemble the key elements of the corticostriatal-thalamic cortical circuit and functionally integrate them into loop assembloids using custom 3D printed biocompatible wells.
Yeah. Yeah, I thought that was interesting too.
So what do they do? Are they doing just how are they building these circuits? I'm not quite sure.
Well, so manipulation, just manipulation of organoids is kind of a microfluidics problem.
Yeah.
Right. And I think this is where you see the this again interaction of the a lot of organ on a chip kind of work, making custom hardware to allow to kind of, because we don't know how to tell cells what to do.
Yeah.
We kind of have to put them in an environment where they just do that thing that we want them to do.
Yeah.
And so it's a combination of where nutrients are, where are the forces, whether you're generating those forces with centrifuges or you're generating those forces with microfluidics. This is, again, you'd see from the paper's figures that it's quite complicated equipment generation, and very, very specialized, right?
Yeah.
And you see this with the you see this in the even the kind of low cost equipment that the Braingineers publish about for quality control of single organoids, right? So this is much more complicated. I think think this might be the largest number of largest assembloid. Like, I'm pretty sure Vienna's still doing 3. Anyway, yeah, but really interesting, and again, they're combining this with all sorts of kind of specialized neuronal techniques as well, right? So tracing and calcium imaging, and it's yeah. This is the kind of stuff where it's just like, oh, okay, we're not going to be able to recreate this.
Yeah.
Low cost way.
Yeah, this is pretty advanced.
There's a lot of skills. It's certainly, you need a team for this, because you need lots of different specialists.
Right. Yeah, it looks like. Yeah, so it looks like they're doing a lot of this stuff to try to sort of recapitulate early human development and then disease conditions.
Yeah.
So that's good. Yeah, and then this is a LinkedIn post here. This is a link to a paper, New Perspective in Cell, where they discuss the concept of AI scientists.
Oh, right, and yeah. Why do I?
Biomedical discovery, I guess.
Yeah, I think this was this was more in there because, yeah, just there's so much you need to know in biomedical research, know, and the utility of these agents. Yeah.
Yeah, I think that's it's a. Of course, we always talk about bioinformatics and trying to put together knowledge.
Yeah.
Looks like they're really kind of moving towards some of the trendier topics like continual learning.
Absolutely, right. I mean, it's I was in looking at some of the software behind evolutionary robotics.
Yeah.
And things that I could, add to the kind of basic Bungard Pi Bullet stuff, made me check out Ida Momenajad's work, because I was thinking about her NeuroNav project, and it was just interesting because it was just like, Ida's now like all large language models.
Yeah.
And this is so, someone, I mean, it's, good for Richard Sutton. He's stuck. He's studying RL and he's sticking with it.
Yeah.
So, anyway, it's again, there's so much. I mean, it's really interesting to me listening to people talking about large language models, coming up with new ways, new creative ways to say, sometimes large language models work and sometimes they don't, and we can't tell the difference. But it's like you keep coming up with new, what did somebody was describing it as like fractal. I don't know. They were trying to give it a new fractal name. It was just one, you're talking about hallucination.
Yeah.
Two, like this new term adds nothing to the discussion.
Maybe I had seen that somewhere, but it sounds weird. Yeah, it sounds like something we want to avoid, I guess. Yeah. Well, anyways, fractal nonsense. I don't know.
Right. I mean, I think he's a force for good, but I think Sean Carroll started it with saying that he's a fractal professor at Stanford Institute.
Oh yeah, well, at least there it makes a little bit of sense because you study fractals.
It does.
So yeah, This is the Weiss Lab. This
Yeah,
is a link to
I dropped that in terms of a link to the tissue talk.
Ron Weiss, yeah. This is more from Sergio Pasca on organoids and assembloids.
Like I said, he keeps banging on these things.
Oh, so this is an image of the last paper we talked about, right? Is this these are the so you have the pluripotent cells building these diencephalic organoids, doing single cell RNA-seq, and then growing them for 100 days and seeing the characterization of the different cells there. And this is a UMAP plot of the cells and their markers. And then this shows this is another UMAP showing the different cells in the organoid. Yeah, this is really interesting. And then you have spatial mapping here. So it corresponds to E15.5 mouse. Yeah, that's interesting.
Yeah. No, it's great stuff. And yeah. That is coming off of, I think, off of a James Glazier well, anyway, but certainly of the kind of CompuCell 3D kind of simulations.
Yeah, this looks like agent-based modeling, PhysiBoSS.
Yeah.
So this is multiscale models. Is there a paper associated with this?
Don't hit the paper. I mean, don't hit the image. Hit the LinkedIn link.
Oh, I don't I don't like to get into LinkedIn in the.
Oh, okay.
Could you paste the paper if you could?
I will try. Sorry about that.
That's okay.
I mean, these days it's just like I just don't like to use Twitter.
Yeah, I know. Avoiding that like the plague.
Let me find that. Yeah. It's so it's so hard to find. Why can't we all just use web links?
Yeah, I know. It's like.
Okay. Oh, okay. There's quite a lot of stuff here.
Yeah.
Here we go. Let me see what he's got. Yes, PhysiBoSS tutorial. Here it is. I mean, the other thing that I feel bad about is the is to go in, extract the paper, right?
Yeah.
And then like kind of bypass this person's post. You know what I mean?
Yeah, I know.
Anyway, there you go.
That's okay. Yeah. So this is building multi-scale models of PhysiBoSS and agent-based modeling tool briefings in bioinformatics.
Yeah. And this is the kind of stuff where I'm really hoping, like again, like there's this third leg to the kind of I don't know if I want to call it the Organoid Intelligence Project or kind of Organoid simulation project, but how to bring much more sophisticated biological simulations to play that like again, like the really nice thing about the Vanguard paper is that he did it and got sim to real.
Yeah.
In that, like, okay, now if I make that, does it behave the way that my simulation said it would? And so yeah, I thought that was really I'm always looking for things that are in the kind of CompuCell 3D Morpheus space or Tissue Forge space. Yeah.
This of course is just kind of in that spirit. So this article introduces 3 examples of multi-scale models which rely on the framework PhysiBoSS, an add-on of PhysiCell, which includes the intercellular descriptions as continuous time Boolean models to the agent-based approach. The article demonstrates how to construct these models more easily, relying on PhysiCell Studio, the PhysiCell graphical user interface. And then they give a step-by-step tutorial in their GitHub repo here. So this has really kind of working from a Boolean model and building these agent-based models. Okay, so that's great.
And I think like TNF models would be cool.
Yeah. All right, so that's great. Why don't we move on in the interest of time here? We were here. So the next paper is the Systematizing Cellular Complexity, a Hilbertian approach to biological problems. This is in PLOS Complex Systems, which is a newer PLOS journal. So this is Nima Dingani, and this is sort of this idea of, I guess...
Yeah, I mean, certainly trying to, kind of a theory paper, right?
Yeah.
That's harkening back to Hilbert's, 1900 problems in mathematics.
Yeah. So this kind of goes through, talking about how, you can examine individual components of cellular systems, and you can find molecular reactions and interactions, basically more of a sort of a reductionist approach to getting information out of the cell. However, the challenge lies in integrating these components into a comprehensive system scale map. And so this is just making this system scale map better and more useful. This difficulty arises due to factors such as missing links, unknown variables, overlooked nonlinearities in high-dimensional parameter space, downplayed natural noisiness and stochasticity, and a lack of focus on causal influence and temporal dynamics. And then, what is it that is the essence of complexity in the cell, basically? So this is, of course, important in constructing a meta-theory of biology, and is addressing fundamental aspects of cellular regulation, adaptability, and noise management is vital for understanding the robustness and functionality of biological systems. So this is all about, not only the full description or a better description, but about some of these other things: robustness, functionality, adaptability, etc. So this is really kind of, he has three problems here. One is where are the control switches. Two is how to manage the need to reconfigure. And three is how to harness noise rather than to succumb to it. So the first part here is this control switches, which is, of course, actually cites here Ashby, where you have the concept of trial and error. Karl Popper highlighted the role of trial and error as a fundamental process in knowledge acquisition within evolutionary epistemology. So, a lot of people propose molecular switches in the cell that switch the cell maybe from one state to another, or switch from one metabolic mode to another. So, what does that mean? Is it something that gets learned? Is it something that's programmed? Is it something that's, a Boolean or binary phenomena, or is it, how do we characterize it? And it has, it relates to adaptation. How can the cell adapt to things? There's been a lot of work on switches and say like E. coli, where, you have a very relatively simple set of switches that can switch the phenotype from one type to another. That's sort of an adaptive form. So like if you're in one set of environmental conditions, you can switch to a certain metabolism, and then if you're in another set of environmental conditions, you can switch to that metabolism. So, it gets into that. Problem 2, how to manage the need to reconfigure. This speaks to homeostatic response and feedback control. And I'm thinking about adaptive reconfiguration in cells. So like from switches to this idea of reconfiguring the phenotype, the metabolic networks and all that. Kind of going through that. And then problem 3 is how to harness noise rather than to succumb to it. Now, there's this idea of cellular noise that's been very popular in the last maybe 20 years. The idea that there's intrinsic as well as extrinsic noise. So you have intrinsic noise that operates at the level of sort of maybe like, you can think of gene expression as having intrinsic noise, having this noise in like stoichiometry of the molecules that are involved in triggering gene expression, and having transcript production that fluctuates over time in different contexts, and how a biological system can handle that noise. So if you had like deterministic gene expression, it would be easy for the organism to know what to do. But when you have this fluctuation, especially over time, the cell has to sort of integrate all that information and say, this is the sort of the signal that's being sent, extracting the signal from noise and all that. And then there's, of course, extrinsic noise, which is from the environment, where you're getting these signals that are noisy from the environment, and the cell has to figure out what the actual signal is and separate that from the noise, so to speak. So this is kind of going through this sort of, talking about noise and how noise is actually good in the organism or in the cell. So, we think of noise as being negative or bad, and the signal being separate from the noise. But sometimes noise is important for biological systems, especially because it provides variety, it provides a means for adaptation and change, it provides information to the cell or the organism that they can use then to understand its environment or understand, kind of understand what's going on in the cell or, in the biochemical network better. So that's getting into that. And then just kind of finishing up, integrating all of these three things into a sort of a meta theoretical approach here. Okay. Yeah, that's good. You want to comment on that, or?
Well, I was just gonna, this is all making me, I'm coming off the back of this reading group, so there's just thinking a lot about Markov blankets.
Oh yeah.
And it's I posted this in the math channel, but the Newton Institute has been hosting a history of mathematics week-long workshop or something, conference, and the is it Revchesky, the kind of very famous biophysicist, mathematical biophysicist in University of Chicago in like the 30s. You know what I'm talking about?
I think so.
Anyway, it's somebody was covering his work, and because he was such an influential figure in biophysics, and was kind of perhaps too zealous in his insistence on the mathematical nature of everything. But that they were covering the fact that he's got this free energy explanation for cell division.
Yeah.
I don't know if you've seen that. Yeah. I've got his. I mean, I think they're in a box now, but I've got his 2-volume biophysics text.
Yeah.
from some I don't know if it's like a Dover publication or something. I haven't gone into it to see whether there's some interesting free energy principle kind of precursor there.
Oh yeah, precursor. Yeah. Well, there are a lot of people doing things with like energetics and
Sure.
But I mean, I don't know. Like I get the sense that like my understanding of free energy principle is basically that it's kind of a rehash in a way of like a lot of the energetics work. Like, you have Prigogine and other people who proposed a lot of things having to do with like energetics and minimizing energetics. And so, I mean, there's that aspect of it. So it has a lot of precursors in early complexity theory. And, now we're talking about like going to active inference and things like that. So, but it's,
again, coming off of that book and I don't,
yeah. So
I don't know whether the book is trying to cover too much. But yeah, anyway, I want to revisit that, or I want to do the class with the Active Inference Institute, or the multi-week reading.
Yeah. So yeah, we have this is a list of regenerative medicine and stem cell journals from the niche.
Oh yeah. So so Knopfler, maybe I was thinking of his name instead of the guy in Vienna, but he's Professor Davis that does the niche as a blog, and like weekly report. Yeah, he's very good.
Yeah. This is
and he actually covers regenerative medicine.
Yeah.
like he's the one who covers all the stem cell clinics, and it goes through the not necessarily valued work of seeing which ones these are just charlatans and things.
Oh, right, yeah. So then this is a bioarchive, or this is a LinkedIn post to a bioarchive paper pointing to a bioarchive paper. An integrative layer resolved atlas of the adult human meninges.
Right.
This is another LinkedIn post pointing to this Cell Reports Methods paper on 3D brain organoid neuronal network analysis strategies report. So this is actually MEA NAP, which is microelectrode recording related. So this is a flexible network analysis pipeline for neuronal 2 and 3D organoid microelectrode recordings.
Yeah. So I wanted to throw that in. The one piece remaining, the one proprietary piece remaining in the Braingineers kind of protocol, or certainly lab automation, is the MEA.
Okay.
And that might be the case for a while. So I was just looking for, who's dealing with the, getting that data out and into an open analysis pipeline.
Right.
And just to have that as on the radar. And what will be more important, of course, for the Organoid Intelligence, right, is like you want to stimulate as well, right?
Right. All right. Yeah, that's important. This is another post to a paper, an integrated transcriptomic cell atlas of new human neural organoids. It's from Nature. And this is the diagram here showing.
Yeah. Yeah, good stuff. And sorry, I'll just link through to see who the so Sergio Pasca again. Here we go. OREL. And I'll just I'll put in the thread there a link to the paper.
Okay.
There we go.
So this is the paper and integrated transcriptomic cell atlas of human neural organoids. This is human neural organoids. It's unclear which parts of the human brain are covered by existing protocols, and this has been difficult to quantitatively assess organoid variation and fidelity. So these 36 single-cell transcriptomic data sets spanning 26 protocols into one integrated human neural organoid cell atlas totaling more than 1.7 million cells. They've cited 26 papers here.
I mean, that's awesome, right? I mean, it's a drop in the bucket.
Yeah.
but it's great to see, or it's a great start, and it's also just I think something that is also really important is this idea of how clear it is what cells are.
Yeah.
So there is this reference atlas that Allen Institute, for instance.
Yeah.
But it's also just interesting in terms of the kind of criticisms that have been made about how normal these cells are.
Right.
Put that together with this issue of there are cells that it's not clear what they are in the brain in a normal human, right? And again, back to this idea that there's a cell type as opposed to it being kind of a spectrum,
right.
And so so just it definitely looking forward to tracking that in terms of how many organoid cell types have been kind of cataloged and
so they say here mapping to developing human brain references shows primary cell types and states that have been generated in vitro and estimates transcriptomic similarity between primary and organoid counterparts across protocols. So they have these exemplars from the developing human brain. I guess from the human adult, I don't know what their data sets look like, but basically they have this ability to map this to things you see in the brain and you can characterize, and that's basically what the goal is here. So it's I guess the whole aspect of the discrete cell states. Sometimes it's kind of hard to know, what a specific cell type is. You get a lot of variation in terms of what they look like, what they express. So it's nice to have an atlas where you can at least have some comparison in a in vitro setting, because in the in vitro or an in vivo setting, because in the in vitro settings you often, get things that are not, seen in nature, basically. So, okay, that's great. Thank you for that. This is the Causal Bench Challenge, a machine learning contest for gene network inference. from single cell perturbation data.
Yep.
This is 5 questions with Professor GLU, Harvard bioengineer and AI and brain-machine interfaces using technology to treat disease. Oh, this is the synthetic biology intelligence lab paper here.
Yeah, right before the holidays.
Yeah. This is endogenous gene tagging with FNCAS9 to track and sort neural lineages from 3D cortical organoids. Yeah. That's interesting.
Sure.
Deciphering brain organoid heterogeneity by identifying key quality determinants. That's like cell culture sort of. Yeah. And then that's and then today's papers that we talked about earlier. So any of these other ones that we kind of highlighted, would you like to get into, or?
I mean, to do the last one, the quality.
This one here,
yeah. So, I mean, again, just super important to, there's this real issue of quality control,
right?
And together with what do we mean by quality control,
right?
And what's important for biological systems? So and being able to understand the difference, or so I'm always on the lookout for papers discussing it, as well as just how which of these techniques would be kind of reachable with a smaller budget.
Yeah.
And so this is just bringing up some of the
easy to assess things.
Yeah.
So this is where they use a ferrite diameter, and that's shown in this figure. And then this is emerged as a reliable single parameter that characterizes brain organoid quality. So you start with like measuring some morphological characteristic, and then you go to the expression profile and you see what that looks like. So that's interesting.
And again, it's want to think about. I mean, the biological intelligence paper, or from scratch, setting up a biological intelligence lab from scratch paper covers a lot of the kind of the infrastructure you need, the reagents you need, but it's not getting at the kind of the protocols and again what Madeline Lancaster identifies as a big problem, which is the inability for different research labs to reproduce each other's work. So it's needing to think about this in like we're not tracking something important. It's the we don't know what we're doing. Just how crude things are at this point, and how to move to something that's at scale, right? So that you're not kind of hand hand rearing each organoid, and just hoping to get lucky. But and then obviously also dealing with some of the issues that I think UCSF was one of the first to kind of try to characterize well, mainly to criticize the work from San Diego.
Yeah.
But that gene expression of organoid cells is unusual.
Yeah.
And so they described it as the cells seem stressed,
right,
or distressed, or
they are. They're in a weird environment, and they're being pushed and pulled.
It's an artificial environment with again artificial signals, and and this is where I think it's going to link back to the kind of Baumgart-Levin Xenobot kind of work. Is you're asking for tissue or network, like you want to make predictions about network behavior from cells that are, in a sense, grown in a weird isolation.
Yeah.
Certainly from their expected tissue context.
Yeah.
Right. And so you're giving it growth signals and other kind of expression signal, artificial growth and expression signals.
Right.
Anyway, and it was interesting, the Baumgart Texas Tech Talk that I dropped in there.
Yeah.
You see more about how it's really just kind of a fancy embryo manipulation that they're doing.
Yeah.
Right. So they haven't gotten into the synthetic biology too much.
Yeah.
But I think at a certain point, I mean, like there's something to be said for that just physical manipulation, cell manipulation.
Yeah.
In terms of just what happens when we don't use any signaling. But that is, of course, also super artificial.
Right.
And like obviously you're gonna to build more sophisticated robots, biological robots, you'll start to be interested in cells signaling and things like that.
Yeah. Okay, that sounds great. Thank you. Why don't we move on to some papers now? I mean, we've been talking about papers, but like in another set of topics. So we have this, let's see. Okay, so I actually did want to talk about one more thing before we get to that. I noticed that the Thousand Brains community is pretty active, and they're doing a lot of interesting things.
Oh yeah.
So this is their YouTube channel, the Thousand Brains Project, and they've been posting a number of videos on. They've been having these brainstorming sessions and posting some videos on this. So this is one aspect of their community where they're trying to work out problems in video, kind of like what we do. This is a video. What is the Thousand Brains Project all about? That was actually quite useful because it kind of talks about what they're doing. They seem to be kind of they're trying to emulate the neocortex. They're emulating sort of the layers of neocortex and using those as information processing steps or stages. But I notice also that they're very standoffish in terms of, are we really simulating the brain? I mean, maybe we shouldn't be surprised by that because you don't want to oversell what you're doing. But they're very much in this area of using the neocortex as a model system. We're building an intelligent agent, basically. And so they have a lot of good content on this YouTube channel. You were going to say something?
Yeah. Well, I was just going to say, I think what you mean by that is that they are utilizing a cortical column model.
Right, cortical column.
As being the kind of the unit of the neocortex.
Right.
And yeah. Anyway, it's I think it had a lot to do with their old software project. Like what I thought of as the Numenta open source work from like 10 years ago.
Yeah.
But I think he is. I mean, he's certainly he's aware that there's a lot of other structures in the brain.
Oh yeah, yeah.
and but that's interesting. What's the community? I mean, is it more than is it more than them?
Well, no.
Are they reaching out to other? Yeah.
From what I understand, it's them. They have a couple key people in the organization, and then they want to get people. They want to recruit people to use their approach. So yeah, they're using the cortical columns as sort of like the fundamental units of cortical processing, and what the idea is you array these columns like they are in the neocortex, just kind of in parallel. You're able to build, these basically parallel computing devices. So they have a YouTube channel. They also have a website here where they have their community tab. So they have they're doing things like building issues on GitHub. They have issues for building on the Monty. We talked about Monty before.
Yeah.
And so there are people doing doing pull request comments and things like that on this.
Okay.
Yeah, so the community is just basically a bunch of people who want to build these biologically inspired intelligent systems based on 1,000 brains theory. So it's kind of like the theoretical side of like Monty and Numenta and all of that, because they've been developing these tools for a while. Yeah, so this is their website with the community tab. And then they actually have recently, I think over the holidays again, put out this technical paper, and it kind of highlights what they're trying to do with 1,000 brains. So this is Viviane Clay, who's working at Numenta. She's a key person in this community. Jeff Hawkins, of course, at Numenta. He's another, of course, one of the key people at Numenta. And then Niles Ledholm, who's also at Numenta. So this is their new paradigm for sensory motor intelligence. Basically, they're kind of they're talking about deep learning, how this has driven a lot of progress in artificial intelligence. Despite these advances, the creation of intelligent systems that can operate effectively in diverse real-world environments remains a significant challenge. In this white paper, we outline the 1,000 Brains Project, an ongoing research effort to develop an alternative, complementary form of AI derived from the operating principles of the neocortex. We present an early version of 1,000 Brains, or the 1,000 Brain System, a sensory motor agent that is uniquely suited to quickly learn a wide range of tasks and eventually implement any capabilities the human neocortex has. Core to its design is the use of a repeating computational unit, the learning module, which are these cortical columns where you have different layers and a column, and then you put these columns into parallel, and you're able to use that as the repeating unit of computation. And you might argue that that's not really realistic characterization of what neocortex actually does, because there's diversity across neocortex. As much as those units repeat, you get different sensory cortices, you get integration cortices, multisensory cortices, and they're, the way that the processing, what's being processed, of course, is different. They're trying to make a generic version of these columns so that, they're not like beholden to any one sensory system. Nevertheless, in neocortex, we see these segregated regions of, devoted to different sensory systems: visual cortex, motor cortex, sensory motor cortex, auditory cortex, etc. So it begs the question then: are they really being used in the same way, or is this something that's very highly generalizable in the way that they're proposing? But anyways, it's it's a learning module that allows you to use this sort of columnar organization. Each learning module operates as a semi-independent unit that can model entire objects, represent information through spatially structured reference frames, in both estimates. And is able to affect movement in the world. So it's basically taking in sensory information, outputting some sort of motor response or sort of response to the world. Learning is a quick associative process similar to Hebbian learning in the brain, and leverages inductive biases around the spatial structure to the world to enable rapid and continual learning. Multiple learning modules can interact with one another both hierarchically and non-hierarchically via the cortical messaging protocol, which is something they've used in Numenta, creating more abstract representations and supporting multimodal integration. So they're really combining cortical columns and then this messaging protocol, which is, of course, important for the computational aspect. We outline the key principles motivating the design of Thousand Brain systems and provide details about the implementation of Monty, our first instantiation of such a system. This is the code here on GitHub, and then detailed documentation on their Thousand Brains project wiki, I guess, or whatever, README site. So this just kind of goes through the details. This is actually, they talk here about the need to develop world models. So this allows the system to quickly learn the structure of the world and how to manipulate objects to achieve a variety of goals, which is sometimes referred to as a world model. So I find it interesting they're going to world models here as well as a lot of other people. So they argue that the Thousand Brain system is built with embodied sensory-motor learning at its core. This is, again, kind of the claims. And
again, like, not taking anything. Sorry, my phone is it's trying to send voicemails for me. But this is very like their I feel like this is a rebranding of what they were doing. I forget what their old project was called.
They had Numenta, and then they didn't really couch it as Thousand Brains. They couched it as kind of like the
right. Yeah. Well, then he had a book.
Right? Oh yeah.
I mean, and that was, again, this isn't to take like at both the Redwood Institute and then what he was doing together with sorry. I'm trying to think of the guy who ran their open source project.
Yeah, I remember him. Yeah.
I can see him, but
Yeah,
I
I
can't
know.
remember his name. Anyway,
That was pre-pandemic, and he died
right.
yeah. So I guess they're trying to revitalize the community and rebrand it a bit, trying to get people to use it.
Yeah. I mean, again, this isn't to take anything away from it, but this is. I do feel like this is very similar in kind of a concept and approach to what they had been doing, and I think in the like the 2 months ago presentation that they did, I think he brought up, because it was like temporal hierarchical.
Oh, temporal hierarchical learning, yes.
Right. Like that was kind of, he was definitely trying to say, like, we're doing something more general and flexible.
Yeah. Well, I think that's kind of the generic version of what or the technical version of what they're doing.
Yeah.
And then this is kind of like, we want to, because he did write a book on 1,000 Brains. I don't remember what it was called, but that was his second book. And so I guess maybe this is more of a more accessible way to kind of approach it. But yeah, I mean, this is fascinating work. I think, they're really kind of working out. I don't know what the difference is between the two. Really, I mean, they have some things here where they talk about the building blocks of what they're doing. Basically,
but I think it gets back to this issue of because Ida Momenijad's NeuroNav project is kind of this intersection of, I'm doing reinforcement learning, or like a model-free reinforcement learning for a simulated agent, but I want something that's neurally plausible.
Right.
And it's a strange tension, because to some degree the neurally plausible doesn't matter.
Yeah, I know. What does that mean? Like, is it like it could happen in some universe, or is it like really hew directly to the brain? What does that mean?
And it's just like, do you care about the behavior, or just that tension?
Yeah.
And and it comes up a lot in, I mean, especially if you want to try and make it into engineering, right? Like neuromorphic computing, where it's just like, do I want, how much do I want the neuro, and how much do I want the computing?
Yeah, I think that you can see that in there, like kind of the highlights of, like, if you look at the list here, you have sensory motor learning and inference. Of course, that's something that is behavioral, but you're generating motor commands. Could you use that in like something like a Xenobot, or could you use it in something like a embodied robot versus like just generating some motor commands that are, and then you have modular structure. Of course, a lot of things are structured in a modular way, not just the brain, but that's cortical messaging protocol, which is a computational abstraction, which, I don't know how closely that is aligned to the biology of the brain, but we have computational neuroscience, which predicts that, there are different types of computational things that the brain does. There's voting, which of course is something where neurons and different units are participating in collective behavior. Reference frames, which of course we know from from neurobiology, is something that we see in the brain and basically has a behavioral consequence. Rapid continual learning, where learning and inference are closely intertwined. So this is again like where you're characterizing the learning process.
Yeah. And then model-free, model-based policy, right? I mean, it's you know, it's comprehensive,
right?
like those are all the things that I want to follow.
Yeah.
I mean, I almost want to say that the cortical message passing is kind of like the active inference secret sauce.
Yeah, okay.
Right, where, because a lot of the Bayesian mechanics arguments for, or a lot of the arguments about like this allows us to put everything in one framework, is the argument for the cortical message passing. Right. That like there is this lingua franca of multiple systems, but that they can then eventually come together. I forget what section of the book that is, but where it's just like this is a way. I think Thousand Brains and active inference people both want some sort of cross modality integration.
Right. So I think that's an interesting exercise in what we might look towards some sort of bio-inspired platform, at least. And again, they're doing this sort of dynamic continual learning, which is it's a nice goal, and it matches everything with with embodiment and that. But I mean, I think this is worth thinking about more, especially getting into maybe the software and working out some examples. I don't know if people have an interest in doing that, but if we do, then we should follow up on it.
I mean, it would be good to check in.
Yeah.
And, like again, I love that list, right?
Yeah.
Continual learning's got to be in there. And understanding, it's trying to bring in anatomy, or it's trying to bring in the different brain structures. It's trying to bring it, the hard things for robots to do is still picking up a phone. Anyway. We'll see.
Okay. So the other thing I want to talk about is this paper. This is a new paper. This is PLOS Computational Biology.
Dan Goodman.
Yeah, Dan Goodman and colleagues. This is Adapting to Time, Why Nature May Have Evolved a Diverse Set of Neurons. So when we're talking about the organoid cultures and things like that, we talked about cell types and validating organoid cell types with what we see in vivo brains. And then, this is where they're considering why we have different types of neurons. We're a diverse set of neurons in a neural network. If you look to the, the cortical networks that we were talking about earlier as well, where you have different structures, the neocortex, the striatum, and the thalamus, they're all kind of connected in the mammalian brain. Why do you need that kind of a circuit? Why can't you just take cortical columns and put them together directly from the brainstem? I mean, why do you need all these intermediate structures and these loops? So that's kind of maybe what they're answering in this paper. So the abstract reads, brains have evolved diverse neurons with varying morphologies and dynamics that impact temporal information processing. So their focus is on temporal information processing, or put in another way, it's continual learning, because you're learning about time, but you're also learning over time. And processing the temporal component is important. In contrast, most neural network models use homogeneous units that vary only in spatial parameters, such as weights and biases. So if you have something like a connectionist model, you have these units. The units are all basically the same. They aren't differentiated except for their weights and maybe their bias. So they're like purely computational biases and variation. It's not, in terms of what the cells are. So, you can have, there's no differentiation, say, between interneurons or, pyramidal neurons or anything else that, any classification we can think of that has some functional differentiation. So this functional aspect is, I guess an important thing to kind of figure out why it exists. To explore the importance of temporal parameters, we train spiking neural networks on tasks with varying temporal complexity, holding different parameter subsets constant. We found that adapting conduction delays is crucial for solving all test conditions under tight resource constraints. So it's this conduction delay aspect which is important. And of course, there's this whole aspect of the brain being sort of a temporal difference, or at least the cerebellum doing that. But in the cerebellum, you have this, relatively complex circuit that allows this sort of temporal differencing. But you see this in individual neurons in terms of the biophysics. So basically, it's this ability to sort of gain this temporal information. Remarkably, these tasks can be solved using only temporal parameters, such as delays and time constants. So there are your delay differential equations, Morgan. They come in use here in terms of modeling temporal delays. And so you can solve these tasks that they presented to this network using only temporal parameters such as delays and time constraints with constant weights. So you're keeping the weights constant, the connections between the units. What you're doing is you're changing the delays and the different time constants. So you have, one network that has one time constant, another network has another time constant, and then you might delay within that different connections. And so instead of the weights, you basically have, like a matrix of delays, and that can also be very useful for information processing. Go ahead.
What's the is it like course or conference that Dan Goodman?
It's SUNFA or something. It's like
like spiking
neural networks conference. Yeah.
Right. Yeah, something like that. Yeah.
Yeah, so that's a whole different world. They do a lot of stuff modeling the biophysics of, and then building it into neural networks. So that's what they're doing there. And then some really interesting results in that community. It's not like, I don't know if it's in the mainstream of what we think of when we think of neural network theory. And
well, no, it's definitely because it's focused on the spiking, but yeah, but I can see it being key to this as well as again, it's what it certainly has is the neurally plausible.
Yes.
That this is again, like studying these simulations really does give you a better sense of how neural tissue is probably working.
Yeah. In more complex spatiotemporal tasks, an adaptable bursting parameter was essential. So again, we had this bursting parameter. So this is interesting because this bursting parameter, there's a whole literature and complexity theory on burstiness, which is where you get these events that are like these non-normal, non-linear events that, so you get these bursts of activity. You don't get this constant activity. So when you're doing the spiking, of course, you have this network with, action potentials, and then you have this bursting aspect to it, which lends itself to this sort of complexity theory interpretation. So it makes sense at that level, at least in my mind. Overall, allowing adaptation of both temporal and spatial parameters enhances network robustness to noise. Again, we're talking about biological noise and how you have robustness to this noise. A vital feature for biological brains and neuromorphic computing systems. So this is, again, a bio-inspired thing that is also very engineering friendly. So it's good. Our findings suggest that rich and adaptable dynamics may be the key for solving temporally structured tasks efficiently in evolving organisms, which would help explain the diverse physiological properties of biological neurons. So the answer that they give to why we have different types of neurons, why we've evolved different diverse sets of neurons, is because we need diverse physiological properties. We need to be able to create these bursty network systems that can sort of distinguish, time delays and things like that. Maybe produce different types of time delays. So you might have one type of cell that has one type of time delay, another type of cell has another time delay. And when you network those together, then it's not so much the sort of the Hebbian learning, it's sort of something else that's based on these time delays and how they are able to synchronize. So this is, and then they say in lieu of these, in lieu of connection weights, you can have a matrix of delays, which basically serve a similar function. So and then see if we have any figures in here. This kind of shows what they're doing, the schematic of the framework for this study. So we have this input encoding, which are spike trains. Remember, these are spiking neural networks. Output encoding pair options. These are spike counts. So you get these spike trains and you have the spike counts. Or output encoding pair options, spike trains. So you can actually count the spike trains or the spike counts as the output. So you have the spiking neural network, where you have spike trains as the input. You have spike counts and spike trains as the output. And then you have this somatic potential, which is where you have the soma of the cell, these inputs that, these sort of multiple inputs that come in. There's a synaptic kernel, which, brings in the input, and there's this delay between the input and the synaptic kernel. And then you have this dendritic filtering, which is where you sum these synaptic kernels together as an input into the soma. So the V soma is the V1 plus V2 plus V3. It's this additive contribution. So that's what they're doing in their spiking neural network. They're taking in inputs, and they have basically an integration rule. And then they have this evolutionary algorithm that they're using, which is where they have these initial solutions. They evaluate them. They sort them by loss function. And then they pick the best or the elite loss leaders or the most loss or something. And so the elites are solutions with best performance, lowest loss. You pick the elites out of this selection of solutions. Then you basically use an evolutionary algorithmic approach. You generate children from the elites. You reproduce those elite solutions. And then you have this where you vary this in the children, where you have random vectors. You have 5 children, and you keep doing this to get different solutions. So you're basically improving the solutions from random, and that's how they're doing this. Okay, so I think that's it for that paper. Did you have anything else to say about that, Morgan?
No, bring up the title again. Yeah, I mean, I think it's really interesting, and especially interested in the evolutionary algorithm part of that, just because that's a big part of Josh Bongard's work, and it's you know, we have a lot of cell types because of evolution, right?
Yeah.
Like it's one thing to say like that's actually useful, but it's a bunch of history, right?
Right. It's historically
And
contingent.
yeah, but sorry, I noticed two weird references on the paper. Just if you bring up the title, there's so one, if you look at Karam's institution, it says University of Bristol, Southwest England.
Yeah.
That's not a place. Is it yes, like Bristol is in the southwest of England.
Yeah.
But
Where
that's
is
not
that?
a place. But then go down a little bit and see the editor, who it was. So editor, University of Edinburgh, United Kingdom of Great Britain and Northern Ireland. Like again, like you know, it just seems like there's some weird algorithm that's deciding this,
maybe.
like as opposed to it being real in the
Yeah.
sense that like nobody writes their address as University of Edinburgh and then that weird, I mean that is the proper name for the United Kingdom.
Yeah.
But like, why was it used there? Anyway, sorry. Not totally relevant to the paper. The evolutionary algorithm stuff, I think is interesting, just in how people construct those algorithms,
right?
Which is like basically all of Gecko and things like that, right? Yeah, just it's interesting the games you have to play to get something that is akin to evolution,
right?
And how the games are usually very crude,
right? Well, I think there's a difference between like using an evolutionary algorithm to sort of solve an optimization problem and like actually doing like, well, saying these are evolutionary scenarios which we're going to test. Yeah. Because I mean that's really the maybe the better question. I said, okay, one option is that these networks evolved because they needed this sort of diverse time information. And then another option might be that it's just historical contingency, that you have like different cell types, like we have with the organoids, that sort of they evolved and then they integrated into networks, and then that's what we have. So I mean, you could test those hypotheses using an evolutionary algorithm, by the way, but also it's a more direct test of like the evolutionary imperative.
I mean, this kind of goes back to the CISIC stuff and the what was that reading group that they were doing in terms of comparative vertebrate?
Oh yeah, the comparative anatomy.
Yeah. We'll follow up. I wanted to, again, thinking about like NeuroNav relative to just doing model-free reinforcement learning.
Yeah.
It made me think about like how much do we have in terms of non-human primate comparative imaging, that we could actually look at some of these things and make inferences or get any insights, and that would be interesting stuff to pull together. Again, thinking about kind of vanguard kind of work, as well as like the whole brain emulation kind of work, right? Where it's just like, hey, we can get all this structure. How much does it really tell us? And how much is it telling us that, how much is it telling us that is relevant if we're missing the kind of the evolutionary context in which it developed?
Right.
It specifically in terms of its functionality, or like how it's being repurposed over these years, and like what limitations are there on that repurposing. Anyway, we can follow up. I gotta.
All right.
Good.
All right. Good discussions. Let's follow up on Slack and see you next week.
Sure. All right. Take care.
Take care. Bye.
Bye.